Hi, my name is Catherine Elliott. The research that I'm presenting provides a new method for the synthesis of macrolide antibiotics. In order to fully convey the need for this new method of synthesis, I'll first begin by briefly explaining the origin of macrolide antibiotics, as well as highlighting their trends in the antibiotic market. The two different types of macrolide antibiotic synthesis, semi-synthesis and full synthesis, will then be able to be explained. Then I'll talk about why full synthesis is better suited for future macrolide synthesis. In 1949, erythromycin, which is a commonly used macrolide antibiotic whose structure can be seen on the right, was discovered in the Philippines. Following the discovery of this naturally occurring antibiotic, other macrolide antibiotics were able to be produced through a process called semisynthesis, where structures called R groups are chemically modified in order to increase the efficacy of an antibiotic or where in the body the antibiotic can be used. This can also help target certain types of bacteria. For example, erythromycin cannot be used for bacterial infections in the gastrointestinal areas of the body, but with modifications to R groups, azithromycin can be made. Azithromycin is used to treat bacteria in the stomach. Modifying R groups or even the carbon skeleton of erythromycin through the semisynthetic method has produced 5 other antibiotics: azithromycin, clarithromycin, telithromycin, cethromycin, and solithromycin. However, the latter 2 are still in the trial stage, and other antibiotics experience significant resistance from bacterial strains. Although other proposed modifications to erythromycin structure would prove effective against bacteria, these modifications have been unable to be made due to limitations in the semisynthetic process. The slow production of new antibiotics poses a problem in light of the increase of bacterial resistance to antibiotics that has been brought about by the overprescription or general overuse of antibiotics, as well as patient failure to finish an antibiotic prescription. When this happens, bacteria are not fully killed off and are able to reproduce with a new resistance to the antibiotic that was originally used to treat that bacteria. So why is the production rate of new antibiotics so slow? First, we have to understand semisynthesis. The starting materials used in semisynthesis are naturally occurring antibiotics such as erythromycin. Modifications are then made to R groups on that antibiotic in order to change the antibiotic. While originally using erythromycin as the starting material was ideal because simple modifications could be made, recent proposals for new antibiotics have either required many steps to produce or have been impossible to synthesize. The difficulty arises because there's no way to target a specific R group. Extra steps can be added to the synthetic process to make a chemical reaction more likely, but in many cases there's no way to ensure the production of a desired product. For example, when producing clarithromycin, this OH group needs to be modified so that it becomes OCH3. However, there's also an OH group here, here, and here. The more complex the modification is, the more steps that are added, and the more likelihood that the reaction is altogether impossible increases. The problems that are seen while using the semisynthetic method can be solved by instead using the full synthetic pathway. With full synthesis, simpler starting materials are used as building blocks to produce the antibiotic end product. This allows for a more simple synthesis that can produce very diverse antibiotics. While full synthesis does require quite a few steps, the number of steps is still less than the number of steps for semisynthesis of recent antibiotics. One drawback of the fully synthetic pathway is the final step of macrocyclization, which forms the ring. This step is difficult to achieve consistently. The fully synthetic pathway works much like a puzzle. Imagine that each one of these beads It's a different section from erythromycin. Erythromycin would be made by combining all 8 of these starting materials together one by one, and then ending with the synthesis forming the ring. If any modification needed to be made, the specific R group that needed to be modified could be targeted, and then modified before it was added to the chain. Then you could pick exactly which part of the antibiotic you wanted to change without worrying that you would change another part as well. This way of synthesizing multiple medium-sized molecules from small building blocks and then combining them to form large molecules is called a convergent synthesis. The convergent nature of full synthesis allows for its diverse products through its ability to target specific R groups. The fully synthetic process has already been used to produce 300 different macrolide antibiotic candidates, as well as produce telithromycin, a current macrolide antibiotic, and telithromycin, which is a clinical candidate. The swift development of new antibiotics has been achieved using the fully synthetic process, proves that the fully synthetic process is a successful platform and can be used to synthesize many new macrolide antibiotics in the future.